Doctors can now edit human DNA, fixing faulty genetic mutations that once caused lifelong, incurable diseases. Welcome to the age of personalized medicine. CRISPR acts like biological word processing, allowing scientists to find, cut, and rewrite specific DNA sequences. Discovered originally in bacteria, this natural defense mechanism protects microbial cells against invading viral genetic material. The Cas9 enzyme pairs with guide RNA to locate and cut exact sections of faulty DNA. Personalized medicine customizes gene therapies to match the exact genetic profile of an individual patient. Thousands of rare inherited disorders, previously deemed untreatable, now have direct molecular targets for correction. Treating sickle cell disease marked the first approved CRISPR therapy, transforming patient lives worldwide through science. In vivo therapies inject genetic editors directly into patients, targeting specific organs like the liver. Ex vivo therapies extract patient cells, edit them inside clean laboratories, and infuse them back. Lipid nanoparticles and engineered viral vectors carry molecular editing tools safely into targeted human cells. Base editors change single DNA letters without double-strand breaks, greatly reducing the risk of cellular damage. Prime editing allows precise insertions and deletions, expanding the range of treatable human genetic mutations. Engineers program human immune cells using CRISPR, training them to aggressively hunt and destroy cancer. Scientists rigorously test therapies to prevent accidental off-target cuts that could trigger unexpected genomic side effects. Patient immune systems must be monitored to ensure the body does not attack foreign Cas proteins.
Hundreds of clinical trials are currently testing gene therapies across cardiovascular, genetic, and metabolic disorders.
Specialized cleanroom facilities manufacture patient-specific gene edits under extraordinarily rigorous, sterile pharmaceutical conditions and standards. Individual CRISPR treatments cost millions of dollars, making affordability the next critical bottleneck in medicine.
Global scientists maintain strict ethical guidelines to ensure gene editing remains focused exclusively on curing diseases.
Therapies only alter somatic cells, ensuring genetic edits help patients without passing modifications to future generations. Artificial intelligence models now predict ideal guide RNA sequences, optimizing editing efficiency while minimizing molecular errors. Leading biotechnology hubs across North America, Europe, and Asia race to commercialize next-generation genetic therapeutics. Developing standardized manufacturing methods will be crucial to bring life-saving gene therapies to patients globally.
Personalized gene editing is transforming medicine, turning once fatal genetic conditions into curable medical realities.